renal corpuscle or Murphysian corpuscle. The renal corpuscle is spherical or slightly flattened in shape, and the diameter is 200 mu. Here you can see the structure of the nephron. This one is the afferent arteriole, efferent arteriole, glomerulus, Warman's capsule enclosing the glomerulus. This is the proximal convoluted tubule, then the thick descending segment, the descending segment, and the ascending segment, then the thick ascending segment and the distal convoluted tubule, and finally the collecting duct. Then come to the function of the renal corpuscle. Its function is to filtrate the blood. The situation of renal corpuscle is cortex of kidney, either near the periphery or near the medulla. Then what is the classification of nephrons? Nephrons are classified into two types based on the situation of the renal corpuscles. The two types are cortical nephrons and juxtamedullary nephrons. What are the cortical nephrons? The cortical nephrons are the superficial nephrons, and the juxtamedullary nephrons are they have corpuscles in the inner cortex or near the medulla region or corticomedullary junction. But the cortical nephrons have the corpuscles near outer cortex or near the periphery, and most of the nephrons are of cortical type. That is 85% of nephron are cortical and 15% of nephron are juxtamedullary nephrons. Then come to the structure of the renal corpuscle. In the structure of renal corpuscles, this is divided into two parts, that is glomerulus and another is Bowman capsule. Let's come to the glomerulus. I have made this in a schematic format which will be easier to understand and write in the examination. Glomerulus is tuft of capillaries and it has efferent arteriole and afferent arteriole. Efferent arteriole is at one end and afferent arteriole is at the other end. Capillaries arose from the afferent arteriole and they divide into 4 to 5 many large capillaries, and those large capillaries divide into small capillaries. Then these small capillaries are arranged in irregular loop form and they form anastomosis. Then again these small capillaries unite to form the efferent arteriole, and we know that afferent arteriole enters the glomerulus and efferent arteriole exits the glomerulus. So after formation of the efferent arteriole, it leaves the Bowman's capsule. And then the efferent arteriole have diameter less than the afferent arteriole, which is physiologically significant properties. Then glomerular capillaries made from single layer of endothelial cells attached to the basement membrane. This one comes under the histology of glomerulus. Glomerular capillaries made from single layer of endothelial cells, and these endothelial cells are attached to a basement membrane. And the endothelium has pores. These pores are known as fenestrae or filtration pores. Fenestrae or filtration pores. The diameter of the fenestrae or filtration pore is 0.1 mu. And the presence of this fenestrae is the evidence that the filtration function is one of the function of the glomerulus. Then glomerulus is over. Then come to the another structure of renal corpuscle, which is Bowman's capsule. It is a capsular structure. It encloses the glomerulus. It has two layers, which is inner visceral and outer parietal. The visceral layer covers the glomerular capillaries, and the visceral layer continues as the parietal layer, and the parietal layer continues as the wall of tubular portion of the nephron. The functional anatomy of the Bowman capsule is like a funnel with filter paper. A funnel with a filter paper, it looks like Bowman's capsule. Then both the layers, the visceral layer and the parietal layer, have single layer of flat epithelial cells. Have single layer of flattened epithelial cells. These are resting upon the basement membrane. In glomerulus, we have seen that it has glomerular capillaries made from single layer of endothelial cells attached to the basement membrane, and here we have the both layers, the visceral layer and parietal layer of the Bowman capsule have flattened epithelial cells with a basement membrane. Then basement membrane of the visceral layer fuses with the basement membrane of the glomerular capillaries, and they form a separation between the glomerular capillary endothelium and epithelium of visceral layer. Then each cell connected with the basement membrane by cytoplasmic extensions of epithelial cells, which are known as pedicels. Each cell in the basement in the two layers are connected with the basement membrane with some cytoplasmic extensions. Those cytoplasmic extensions are known as pedicels. Pedicels means feet, and the pedicels are arranged interdigitally, leaving some space which is known as cleft in between epithelial cells with pedicels. This gap is known as slit pores, and epithelial cells with pedicels are known as podocytes. Then come to the tubular portion of the nephron. The tubular portion of the nephron is the continuation of the Bowman's capsule. It has three parts, that is the proximal convoluted tubule, PCT, loop of Henle, and the distal convoluted tubule. Then first one is the proximal convoluted tubule. It is the coiled portion arising from the Bowman's capsule. It is situated in the cortex. It continues as the descending limb of the loop of Henle. Its length is 4 mm and the diameter is 55 μ. Its functional histology. It is formed by single layer of cuboidal epithelial cells. Presence of head-leg projection is the characteristic feature of the PCT. The head-leg projections are again directed towards the lumen of the tubule. The presence of the head-leg projection, it is the reason that the epithelial cells are called as brush-bordered cells. Then come to the loop of Henle. Loop of Henle has a descending limb, hairpin band, and an ascending limb. The descending limb is again divided into two parts, which are thick descending limb and thin descending limb. The thick descending limb is the direct continuation of the PCT. Then it descends down to the medulla, and it is formed by brush border cuboidal epithelial cells. These are important points regarding the MCQs in competitive exams. And the length of the thick descending limb of loop of Henle is 6 mm and the diameter is 55 μ. We have seen that the diameter of PCT was also 55 μ, and the diameter of thick descending limb of loop of Henle, it is also 55 μ. Then the thin descending limb. The thin descending limb continued as the descending segment. It is formed by flattened epithelial cells without brush border. Again, without brush border and continuous hairpin band. The hairpin band. This structure looks similar as a hairpin, so this part is named as a hairpin band. This is found by flattened epithelial cells without brush border. Without brush border. Then ascending limb. It is also divided into two parts, that is thin ascending segment and thick ascending segment. Thin ascending segment is the continuation of the hairpin band, which is made up of flattened epithelial cells and no brush border. The thick ascending limb, it is 9 mm long and 30 mu in diameter, and it is made up of cuboidal epithelium, and it has no brush border. The thin ascending segment had also no brush border. It has also no brush border. Then it is it's the ascending, the thick ascending segments terminal part runs between the afferent and efferent anteriole and forms the macula densa. And we will read about macula densa in later topics. Then the distal convoluted tubule. We have already seen the proximal convoluted tubule and loop of Henle. The third part of tubular portion of nephron is distal convoluted tubule. Distal convoluted tubule is the continuation of the thick ascending segment. You will see in the diagram of the nephron that we have done here. This one is the distal convoluted tubule, and it is the continuation of thick ascending segment. In the thick ascending segment ascends upward and coiled, then forms the distal convoluted tubule. It occupies ascending segment and cortex of kidney. It is present in the cortex of the kidney, and it continues as the collecting duct. You see here, the distal convoluted tubule continues as the collecting duct. This one is the collecting duct. Yes, the total length is 14.5 to 15 mm, and the diameter is 22 to 50 mu. Diameter varies between 22 to 50 mu. And then the functional histology. It is lined by single layer of cuboidal epithelial cells with no brush border. These points I am again repeating them. These points are important, very important regarding MCQs. And epithelial cells of DCT are called as intercalated cells. Then come to the terminal portion of the nephron, that is collecting duct. The DCT continues as the initial collecting duct present in the cortex. Then the lower part of the collecting duct lies in the medulla. Then the initial collecting ducts unite. Certain number of, that is 7 to 10 numbers of initial collecting ducts unite to form collecting duct. The collecting duct passes through the medulla. Then it is formed by the columnar epithelial cells. Then the histology. It has two types of epithelial cells: principal cells known as P cells and intercalated cells known as I cells. We have seen before about intercalated cells. The epithelial cells of DCT are known as intercalated cells. The collecting duct has also two types of cells: principal cells and intercalated or I cells. The length of the collecting duct is 20 to 22 mm, and the diameter is 40 to 200 micrometer. And now nephron may come as a short note, also it may come as a long question. So you have to write according to that in the examination. Now the again a long question of physiology is over. Thank you.